Welcome class, Aura Bollinger here. I'm going to be presenting malignant hyperthermia to you. This is just an intro. I just want to give you a little background and just to kind of prime the pump a little bit for our lecture next week. In this short video, we're going to be discussing malignant hyperthermia by offering you the definition and giving you a quick background of what it is. We're going to be talking about the incidence as well as the prevalence of malignant hyperthermia in the United States. But really what this video's purpose is to review normal muscle anatomy and physiology. I want to review the normal events in muscle contraction and relaxation, because once you get a good foundation of what normal muscle physiology is, when we talk about the pathophysiology of malignant hyperthermia, it will make so much more sense next week. MHOS, or the Malignant Hyperthermia Association of the United States, defines MH as an uncontrolled increase in skeletal muscle metabolism that's triggered in susceptible individuals by commonly used general anesthetics like isoflurane, desflurane, sevoflurane, halothane, which you'll probably never use, and the paralytic succinylcholine. In other words, when you have an individual that is genetically susceptible to having MH, when they're exposed to these triggering agents, whether it's those gases or the paralytic succinylcholine, their skeletal muscle will go into a hypermetabolic crisis. And so they call it a pharmacogenetic crisis because it was triggered by those agents. And they will clinically manifest as skeletal muscle hypermetabolism. It sounds horrible, and it is, but fortunately it's very rare. It is life-threatening, more life-threatening in the past. Now we've got lots of ways to attenuate it and recognize it. But it is passed on genetically. There are genetic mutations, which we'll talk about, but most likely it's passed on from a parent. And so if a parent has a genetic susceptibility, or if it has this genetic component on chromosome 19 for MH, there's a 50% chance that they will pass it on to their offspring. The exact incidence of MH episodes is actually unknown, but it's estimated to occur in the general population. In adults, about 1 in 100,000, and in pediatrics, 1 for every 30,000 anesthetics. And this is actually probably an underestimation, because a lot of the MH episodes are not full-blown MH episodes in its classic form where you see all of the classic signs of MH. Sometimes they're very mild, they're very subtle. They can be atypical or completely unrecognized, and so then they will go unreported. So this estimation is probably a little bit higher as far as the incidence goes. They have found that it occurs more likely in males than females. I think one of the reasons why is because it's a skeletal muscle issue, and men tend to have more muscle mass, and so they probably have a more profound effect. It happens at a higher incidence in children, less than 19 years old, account for about half of the reported events. And you'll notice the 1 in 30,000, it has a higher incidence in children. And we'll talk about this in our formal lecture, but the reason for this is because the patients that are at risk for developing MH have these underlying syndromes, whether it's a muscular sort of dystrophy, channelopathies, but they have these syndromes that are not apparent until they get older. So they might look like a really normal, say, 3-year-old when they actually have a muscular dystrophy or some sort of muscular issue that usually does not clinically manifest until they're maybe 8 or 10. So a lot of times it goes completely unrecognized in children, and that's why they have a higher incidence of these episodes. Now, because this is a hereditary issue, we do find that the incidence and prevalence is higher in certain pockets in the United States where these MH families, these cohorts, are living in any given geographic area. So we have these MH hotspots. Wisconsin, Nebraska, West Virginia, Michigan tend to be some of the areas where the prevalence of MH is higher, just because this is where the family cohorts are residing. All right, we're starting to get into the good stuff here. What causes MH? What makes an individual susceptible to malignant hyperthermia? Well, as I mentioned, 50% of the cases are inherited, so you're going to get it from your parents. But for those that didn't, it's presumed to be the result of a spontaneous or random genetic mutation. Now, isn't that terrifying? So we could be mutating as we speak. There are well over 100 MH mutations, in fact, but they have found that the most causative mutation, the one that causes the most problems, is the ryanodine receptor, or the RYR1 receptor. It's located on human chromosome 19, and this actual specific receptor has over 700 mutations alone. So that's even more terrifying. But the ryanodine receptor is the specific site that controls or regulates the movement of calcium from the organelle, the sarcoplasmic reticulum, into the intracellular space of the muscle cell. So that's the gate. The ryanodine receptor will control how much calcium gets into the muscle cell and how much calcium gets out of the muscle cell. And you already know that you need calcium for muscles to contract. So you can imagine the more calcium that gets into the cell, the more contraction you have, and the less calcium that gets into the cell, the more relaxation you have. Now, the ryanodine receptor is an important link to MH, but it's not the only one. There's another receptor here, the DHP, or the dihydropyridine receptor, that's also associated with MH cases, but very small proportion, only about 1% of the cases. Here's the ryanodine receptor, and here are the DHP receptors. Now, they're closely associated and coupled with the ryanodine receptor, but it's a separate entity. So these are activated when depolarization occurs, and then it activates the DHP receptors and helps regulate the passage of calcium. So this is the sarcoplasmic reticulum here, and it controls the release of calcium out into the cell. But we'll get more into that. Even though they have identified that this is the issue, or one of the issues, the main issue, I should say, the research is still ongoing. After so many decades, they are still highly researching this. Many great advances since it was first discovered in the 60s, but they still don't know the specific mechanism or the defect of why these general anesthetics and succinylcholine triggers MH. So what triggers MH? I actually just told you. It's the halothene's not listed here, but that would be another inhalational agent. But any of these inhalational agents, isoflurane, desflurane, sevoflurane, and then the paralytic succinylcholine is what triggers MH to occur. An MH crisis is one that is best prevented. So our job is to obviously know what to do if an MH crisis occurs, but this is the thing where you want to prevent it from occurring anyway. We can do a really good job at identifying who's at risk and then just simply not giving them these agents. So I actually want to mention real quickly, just for the record, in extremely rare cases, there is something called awake MH, meaning they don't have to be under a general anesthetic or have the paralytic succinylcholine administered to them. They can have an MH crisis in the absence of those agents. It's called awake MH, and in extremely rare cases, MH susceptible individuals, when they're exposed to environmental conditions that are super severe, like severe heat, intense physical stress, emotional stress, they can go into an MH crisis. So it's important to identify those people and ask them in maybe your pre-op interview, are you sensitive to those extreme conditions? Have you had reactions? And they would know. They actually discovered this with swine. These little piggies would have the same ryanodine receptor defect, and when they were exposed to extreme heat, stress from separation on their way to the slaughterhouse, and even under general anesthesia, they did some experimenting, they realized that these pigs go into an awake MH issue too. And then again, they put them under general anesthesia and realized that it was the same condition that humans have. So there is something called awake MH. It's not just in the humans and pigs, but they've also discovered that it's in some of the little horsies too. Isn't that sad? All right, gang, we're getting into the meat of it, no pun intended. The reason why I brought you here today is to review the normal muscle anatomy and physiology, because if we're ever going to appreciate what happens to the skeletal muscle in an MH crisis, we have to first remember what is supposed to happen with normal muscle relaxation and contraction. Now, as you recall, skeletal muscle has a very orderly arrangement. It's made up of these fascicles, and then within these fascicles are these individual muscle fibers. So we're going to zoom in on this muscle fiber here. Okay, so this picture provides a closer look at the internal structure of a muscle fiber. Now remember, these are actually muscle cells, but because they're elongated, we call them a muscle fiber, but they're muscle cells, and so I'm just going to focus on the anatomy of this cell that's relevant to MH. First, we have the sarcolemma, which is the cell membrane of the muscle fiber. Then there's the sarcoplasmic reticulum, which is the name of the endoplasmic reticulum for that muscle cell. And then these T-tubules that interconnect the sarcoplasmic reticulum. It surrounds the myofibril along with the sarcoplasmic reticulum. And then there's the T-tubules. There are actual invaginations here into the sarcolemma here. So you can see it runs here and then actually dives down deep into the cell and then dives down deep into the cell. So they create a little pathway that goes deep into the cell there. Okay, let's dig a little bit deeper. Here we are with our muscle cell or our muscle fiber, but we're going to pull out a myofibril here out of that, and that is what is considered the contractile unit of the muscle cell. It is made up of two types of filaments, the thin and the thick. The actin is the thin filament and the thick is the myosin, and the arrangement of the actin and the myosin in the myofibril is what gives muscles that sort of striated look. And so this is the contractile unit of the skeletal muscle, and these filaments will contract when there's an action potential that causes depolarization. Okay, speaking of depolarization, let's move on to the neuromuscular junction. So here we are, we have the muscle cell or muscle fiber, and within the muscle fiber, we've got these myofibrils that we just talked about. So let's layer on a motor nerve here that is contacting and innervating a muscle fiber. So we're going to zoom in on this section here, right here. Okay, this slide shows an action potential that's arriving at the end of a motor neuron here at the neuromuscular junction. The action potential then causes an influx of calcium that then causes a release of the acetylcholine into the synaptic cleft. Now down there in step 4 and 5, there is postsynaptic binding of the acetylcholine that causes the sodium to rush in and the potassium to exit, and this creates an electrical charge known as depolarization. Now the electrical impulse has occurred, and that depolarization is spread over the entire surface of the sarcolemma, which remember is the cell membrane of the muscle fiber, and it's carried through the via the T-tubules, which are these invaginations here that will dive deep into the muscle fiber. So the electrical impulse comes here, it dives deep into the muscle fiber, trying to get to the sarcoplasmic reticulum. So that's the T-tubules are sort of the highway. When that electrical impulse reaches the sarcoplasmic reticulum, the reticulum is sort of shocked to release its stored calcium to the contractile unit. And remember, the contractile unit were those myofibrils where the actin and the myosin are held. Now, the calcium that is released from the sarcoplasmic reticulum and now in the muscle cell is available to prepare this myosin head for cross-bridging. The myosin cross bridges bind and pull on the actin filaments, causing the sarcomeres to shorten, and this is what causes muscle contraction. The ATP that's there will help release that cross bridge. Now, after the actin and the myosin sort of do that power stroke and the filaments slide against each other, the myosin returns to its sort of ready state, prepared for the next contraction to begin. This occurs by a reuptake of calcium back into the sarcoplasmic reticulum, and that's what causes cell relaxation. The point that I want to drive home with this is not so much that you need all the details about the actin and the myosin sliding against each other. The take-home message is I want you to understand that this cycle of muscle contraction cannot begin without the presence of calcium to expose those myosin binding sites, and it cannot stop as long as calcium is present. And the entire process requires ATP. So it consumes a lot of energy. As long as calcium is in that muscle cell, the actin and the myosin will continue to contract and slide against each other, and you'll get nonstop muscle contraction, which is why it's so important that the body has a process to have a reuptake mechanisms of the calcium back into the sarcoplasmic reticulum so the muscle has a chance to relax before its next contraction. Okay, let's just quickly review that before we finish up here. So we've got a presynaptic motor neuron here. We've got an action potential that's coming down, causes a calcium influx that then causes the acetylcholine release at the synaptic cleft. When that occurs, there's acetylcholine that binds the postsynaptic motor end plate here that causes sodium to rush in, potassium to come out. That creates an electrical charge called depolarization. Now that electrical charge will travel across the entire cell membrane or the sarcolemma and via the T tubules, which will then go deep into the myofibril and create this release of calcium by the sarcoplasmic reticulum. The calcium that's released by the sarcoplasmic reticulum then exposes the myosin binding sites and allows the actin and the myosin filaments to slide against each other, causing muscle contraction. After the impulse has been received and the muscle has contracted, then that calcium is returned to the sarcoplasmic reticulum via a reuptake mechanism that's in place. And the myosin and actin binding sites or linkages are broken and the muscle returns to its relaxed or ready states or muscle relaxation. Now this entire process of contraction and relaxation requires ATP energy, a huge amounts of energy. And again, the take-home message or the bottom line is that muscle contraction cannot take place unless there is calcium in its presence, and muscle relaxation cannot take place unless there's an absence of calcium. So calcium does play an important role. It needs to be present for contraction, but it needs to be gone for muscle relaxation. Well, that concludes our intro to malignant hyperthermia and a little rapid review of normal muscle anatomy and physiology. I just want you to know that contraction and relaxation are energy-related processes that consume tons of ATP. And now that you know the molecular events that take place with muscle contraction and relaxation, you will appreciate and understand the pathophysiology of malignant hyperthermia next week. So much easier. So hopefully that will help. We just sort of laid the groundwork for that. So I'll let you chew on that for a while before we meet next week. When we meet next week, we'll be talking about the physiology of MH, and we'll be able to just dive right in there. We'll talk about how a crisis will clinically manifest itself, and then we'll talk about how we manage an acute MH crisis. We'll visit who is MH susceptible so that we can identify these individuals in the pre-op setting, and then how we're going to go about administering anesthesia for these MH susceptible patients. Okay, I'm going to leave you all with a YouTube video to watch on your own time before we meet next week. It's Dr. Denenborough, who is actually the individual who rather serendipitously discovered malignant hyperthermia, and who we can thank for all the amazing advances in what we know about malignant hyperthermia today. The video is, maybe 10 minutes or so. I have to warn you, it's a little old school. You know, the audiovisual is not what we are accustomed to, but it's still very informative. It gets the job done. So I will also provide a link to this in the module as well. Go ahead and watch this on your own time. Nothing to really be tested on. It's just background information, and it's very interesting. I will also have some other interesting YouTube videos in the more formal lecture that I'll have you watch as well. So I will see everybody next week, and if you have any questions, please do ask.